Hello everyone, welcome to GenScript 2018 Spring Webinar Series. I'm Claire Zhou, the Marketing Specialist at GenScript. It's my pleasure today to introduce Dr. Yifan Li as our guest speaker. Dr. Li is a senior scientist at GenScript USA. He specialized in the field of metabolic engineering and synthetic biology. He has extensive experience in building and using combinatorial DNA assembly libraries for metabolic pathway optimization. Dr. Li received his PhD degree from Tianjin University, China. He has developed the first application of using CRISPR-Cas9 system for metabolic engineering in E. coli and has several publications in metabolic engineering and ACS synthetic biology. Today, he will share with us his experience in building DNA libraries to explore the combinatorial design space. If you have any questions during the presentation, you can submit them by typing them in the question box that you can see on your screen. We will have a Q&A session at the end of the talk. So with this, I'll pass the presentation over to Dr. Yifan Li.
Hello everyone. Good morning, good afternoon, and good evening everyone, depending on where you are. My name is Yifan. I'm responsible for synthetic biology research and development in GenScript. Today, this webinar's topic is building DNA libraries to explore the combinatorial design space. After this webinar, I believe you could have a better idea on how to use combinatorial optimization to improve the performance of your engineered biosystem. This webinar is structured in 3 parts. In the first part, we will introduce some basic concepts and strategies for combinatorial optimization for your metabolic pathway engineering. In the second part, we will describe 2 real cases to illustrate the strategies for combinatorial optimization and how we can help our customers for this kind of project. In the last part, we will briefly describe our service specifications and our unique advantages to meet your needs. Now, let's start from the first part. Synthetic biologists are a group of very ambitious people. We aim to change the world by building very complicated metabolic pathways or gene circuits for diverse applications, such as for the production of small molecules and for therapeutic applications like killing cancer cells, and in the field of agriculture to improve food production. But we all know that engineering new biological functions is never straightforward. If we build a single version of this circuit, we never know it will be functional as expected or not. In other words, it is very difficult to forward design a biological system. Several factors may have contributed to this, such as the significant lack of a priori knowledge about our engineered system and the intricate interactions of cellular metabolism with its thousands of components and innumerable interactions. Even the parts or modules we used to build the system is not well characterized. The strength of a promoter sequence may not well determined, or it may have different strengths under different conditions. In the field of metabolic engineering, a newly constructed artificial metabolic routes is inherently imbalanced, which can lead to a variety of obstacles, including the accumulation of intermediates, the formation of side products, metabolic burden, and ultimately growth inhibition. So, for most of the time, we need to go through the design, build, test cycles to iteratively improve the function. We firstly design a prototype or an architecture of the system and the strategies to optimize the system. Then we build different versions of the design, or we can call it a library of different variants of the system. At last, we test these different versions to find out the best one. We can acquire some knowledge to guide our downstream optimization, and we usually need several rounds of the optimization to achieve a really sophisticated performance. Basically, there are 2 types of optimization strategy. A classic method is sequential optimization. This is to identify major bottlenecks in the initial pathway design and subsequently removing these bottlenecks individually. Thereby, we can gradually optimize the pathway performance. This can also be called the debugging or debottlenecking process. As can be seen in the left figure, a pathway element is varied individually. You can see the blue curve first, until a local optimum is identified, which is then fixed as a starting point for the optimization of a second element, and so on and so forth. However, this effort requires substantial knowledge about the pathway property and topology, as well as suitable techniques to quantify the relevant intermediate. Moreover, as can be seen in the figure, the sequential optimization procedure very often arrive at a local optimum rather than a global optimum solution. You can see from the figure that the red point identified is actually not a global optimum. Compared to the sequential debugging method, a more advanced approach is combinatorial optimization, in which several pathway variables or elements are diversified simultaneously. This is illustrated in the left figure. Multiple elements are verified simultaneously, which allows to systematically screen a multidimensional space. Although we measured 25 points for both of the strategy, the combinatorial approach clearly spans a more complete design space and reaches the global optimum. You can see the red point in this figure, and this global optimum is not accessible by the sequential optimization approach. I would like to illustrate the combinatorial optimization with a real case, which is from Professor Chris Ward's lab in MIT for the construction and optimization of 6-ACA biosynthetic pathway in E. coli. 6-ACA is, which is short for 6-amino caproic acid, is the chemical building block of nylon 6. Nylon is the most highly produced synthetic fiber in the world. This building block is now produced from fossil-based chemical process, which lead to significant green gas emission. So it is quite beneficial to use bio-based production, which is considered to be a sustainable and green process. The constructed 6-ACA biosynthetic pathway consists of 6 heterologous enzymes. The initial version of the pathway is functional. However, the cell produced only 8 microgram per liter product. In addition, there was 8-fold more side products than the desired products after fermentation. These indicate that this pathway is insufficient in product conversion and are imbalanced in enzyme activity, and thus requires further tuning for improved productivity. To optimize the pathway, the author decided to test the strong and weak expression for each of the 6 genes in the pathway. So they have designed a library of promoters. +1 indicate a strong promoter for the gene, and -1 indicate a weak promoter. If we use sequential optimization approach, we can optimize the expression level of the 6 genes one by one, and this requires 6 rounds of the optimization. But if we perform combinatorial optimization, we were able to perturb the expression level of all 6 genes simultaneously, which is apparently more efficient for the project. The author chose to use combinatorial optimization for the metabolic pathway and constructed a library of 32 different versions of the pathway in which the promoter strengths were combinatorially switched. They also included 3 factors that capture the media concentration, including ferric citrate, vitamin B1, and magnesium iron concentration. In total, 9 factors were optimized simultaneously, so the total library is 2 to the order of 9, which is equal to 512 different combinations. To reduce the initial combinatorial library size and minimize the number of required experiments, the author used design of experiment approach to carefully select 22 combinations for testing. Finally, with a single round of optimization, the author increased 6 ACA production by 5-fold. Another major difference for sequential and combinatorial optimization is that building a combinatorial DNA library requires very efficient DNA assembly platform. For sequential optimization, we usually test less than 10 constructs at a time and need to change only one part at a time. This is really time-consuming. For combinatorial optimization, we want to be able to test hundreds or even thousands of constructs in parallel, and we want to be able to change all the components across the whole construct. To do this, gene synthesis and traditional cloning alone is not enough. You really need a way to assemble faster and cheaper. There are some commonly used methods for multi-fragment DNA assembly. They all have their advantages and intrinsic limitations. Take the Golden Gate assembly, for example. It is based on type 2S restriction enzyme cutting and ligation, and it is very efficient to assemble more than 5 fragments. However, it has sequence limitations. You cannot assemble a fragment containing the relevant enzyme cutting site in the sequence. Gibson assembly is also very commonly used, and it is based on in vitro homologous recombination. It has no sequence limitation, but the efficiency drops significantly when assembling more than 5 fragments. In addition, it is relatively expensive and low throughput. To really facilitate this kind of assembly project, we have developed our own proprietary assembly platform. It is very high throughput, able to perform hundreds or even thousands of assemblies in parallel. It has no sequence limitation, so you can add any enzyme cutting site for your downstream applications into your construct. It is free of scar, no extra sequence added into your construct. And finally, it is very efficient. We can assemble up to 10 parts in one round of assembly. So how can our platform help with the combinatorial optimization method? I will show this in the next part. Now let's get into the second part. In this part, I would like to show 2 cases to illustrate how to design and implement a combinatorial optimization project. In the first project, our client has constructed a 4-gene metabolic pathway in yeast cells to convert a substrate to a precursor of a small molecule drug. The idea is simple. They put 4 different pathway genes under the control of 4 constitutive promoters and put the whole pathway in a low copy number plasmid in the yeast strain. They got a product, but they are not very satisfied with the titer. It is far from sufficient to reach the commercial production level. Now they want to optimize the pathway to improve the efficiency. A good starting point is to optimize the enzyme for each of the reactions. Varied enzymes from different species may have different properties. If you test different enzymes, you may find a better one with improved efficiency or less product inhibition, which is really valuable for improving the metabolic pathway flux. Our client blasted the database with the original sequence and search literatures, and finally got 50 homologous enzymes for each of the original enzyme. So the plan is to test these different versions of the enzymes to find out a best combination. With the concept of combinatorial optimization, we want to test the variants for the 4 enzymes simultaneously rather than test them one by one. So the design is to randomly insert these 50 variants in each of the CDS position in the pathway. So the theoretical library is more than 6 million constructs. How can we explore a library larger than 6 million? To answer this question, you need to firstly think about the throughput of your screening platform. If you are lucky to have a relevant high-throughput screening method for improving the pathway efficiency, you can explore a very large library size by directly test a pooled library. For example, if your pathway helps your strain to utilize a substrate for growth. You can transform the pooled library into your host and use serial transfer to select the fast-growing strains. However, for most metabolic engineering and synthetic biology applications, very likely you do not have a high-throughput screening method for your interested traits. Actually, chromatography has been the primary testing method in the field. It is nearly ubiquitously applicable in small molecule detection and quantification. The assay is inherently low throughput, so the screenable library size for chromatography are generally limited to less than 1,000 variants. If this is the case for you, a more practical solution is to test a subset of the library by randomly picking different combinations. The initial step is also to assemble the library as a pool. Then, before testing the library into the producing host, we can firstly screen and isolate positive plasmid in E. coli. For this pathway optimization project, we randomly PCR tested 2,000 colonies, and among them, 1,600 have all the desired inserts, indicating around 80% positive rate. We then used the high-throughput plasmid mini-prep method to isolate the plasmid, which were subsequently transformed into the producing host for chromatography quantification. The point of this strategy is that we do not assemble and sequence all the combinations in the library, so we can save a lot of money and effort. It is also unnecessary to know which enzyme version is inserted for every construct, because for most of the time, we are only interested in the genetic information of the top pathways rather than the pathways with only average performance. Also, testing 1,600 clones rather than the whole library is relatively practical for chromatography screening. So we can firstly screen the library and they sequence only the top variants to acquire some knowledge for the downstream applications. For the downstream optimization, after the first round of design build test cycle, we can perform additional rounds of optimization to fine-tune the parameters. To test the diversity of our assembled library, we randomly sequenced 48 clones. We found that different enzyme variants are randomly distributed, indicating very little bias from the DNA assembly. In addition, all the sequenced clones are different from each other, indicating the library diversity is 100%. Our customer transformed the isolated plasmid into the producing host, performed high-throughput fermentation, and used chromatography to analyze the performance. They acquired some variants with significantly improved efficiency. The combination of the enzymes from the top 24 variants are listed here. As can be seen, some of the enzyme variants, the green enzymes in the figure, were highly enriched in the good performing clones, especially for enzyme B and enzyme C. Take the enzyme C, for example. The number 5 and the number 11 variant appeared very frequently in the top 24 clones. For enzyme D, no particular enzyme were enriched, indicating that this enzyme may not be critical for the performance of the pathway. That's all for the first case. Now let's get into the second case. In this case I'm showing here, is for the construction of a gene circuit for a therapeutic application. This circuit is designed to produce a functional protein in response toward a particular environmental signal. It is not an easy job. The dose response curve for different regulators and the expression intensity of a certain promoter may affect the performance of the circuit. So your initial version of the circuit is very likely to be non-functional. For example, the expression level of a regulator gene may be too strong, so the circuit is always active even without induction. Or maybe the regulator binding is too strong to the promoter, so the expression level of the protein is very low even when desired. Either way, you cannot predict the best solution for your design. To optimize the circuit, our customer wanted us to use promoters with different strengths to tune the expression level of 3 genes. They choose 5 promoters with strengths from very weak to very strong to express the genes. In addition, they want to test different versions of the sensors and regulators with different kinetic properties to optimize the circuit behavior. The theoretical combination is 1000, and they want to test every single version for the circuit in the library. For this project, we used our high-throughput assembly platform to assemble all designed 1000 combinations in parallel. We then used high-throughput transformation, automated plating, and high-throughput colony picking to screen for the correct assembly. We acquired 943 perfect clones in the first round and assembled the rest 57 clones in the second round. All the clones were sequence verified, so the customer can test their design directly receiving the library. The key point of the strategy is that we build and test every construct in the library, which can help you to thoroughly explore the combinatorial design space. We optimized the circuit, exhibited stronger and more prompt response toward the stimulus. As can be seen in the left figure, the initial version of the circuit showed very little response to the stimulation. In contrast, the optimized version showed a very clear responsive curve and a very short response time after induction. Particularly, they found that the signal dose responsive curve is extremely sensitive to the strength of promoter 3, which should be very carefully tuned to arrive at the desired behavior for the circuit. So, in the second part, we have introduced 2 projects for combinatorial optimization. In the first project, we construct the library as a pool and randomly picked clones for downstream test. It is like taking a random sample for the whole library, so we call it representative combinatorial DNA library. This library is best suited in the initial phase for your optimization because you can build a pooled library with relatively large size and they take a small sample for the testing. It is just like a quick glance for the whole design space. In the second part, we build and test every construct individually, so we call it arrayed combinatorial DNA library. This kind of library can help us very deeply explore the design space and is thus suited for optimizing nonlinear systems. The number of variants can be tested with arrayed library smaller than the representative library, since it is more costly and labor-intensive to build every construct individually. If the total number of combination in your library is more than 1000, we suggest that you use design of experiment to reduce the library size. Now let's go with the third part, in which we will introduce our combinatorial DNA assembly service. Here at GenScript, we provide a highly customizable approach for accelerating the building phase of your metabolic pathway and industrial microengineering process. With more than 15 years of experience in gene synthesis and our proprietary assembly platform, we can seamlessly assemble inserts with up to 15 kilobase in length and with up to 4 variable slots. This platform is best suited for metabolic pathway and microbial strain engineering, as well as gene expression regulation studies and protein engineering. We provide 3 different combinatorial DNA library services based on your research needs. For high-throughput screening, we have pooled combinatorial DNA libraries. All gene variants are either synthesized or amplified using the templates you provide. Then, they are assembled and cloned into your desired vector. The total plasmid pool contains plasmid up to 10 to the order of 8. We then PCR verify more than 48 clones to determine the percentage of positive clones with the insertion. In addition, we will sequence verify 24 positive clones to test the diversity of the pool. We guarantee that you will have more than 85 diversity in the pool to ensure maximum screening coverage. In addition to our pool library, we also provide representative libraries. These are randomly selected subsets of plasmid from the pool library, most suited for screening 100 to 10,000 combinations of variants. Each construct is individually PCR verified and delivered. Similarly, similar to the pool library, we will randomly pick 24 clones to test the pool diversity and guarantee a minimum of 85% diversity. Again, the assembled insert can be cloned in either our default vector or your vector choice. One important thing to note is that this library format do not guarantee the existence or selection certain exact sequence combinations in the library. If you have exact combination of variants that you want us to synthesize or test, we will recommend our arrayed combinatorial DNA library. For testing every single design in your pathway, we provide the arrayed library platform. Based on your specific request, we will assemble each individual construct for you. Each construct is then PCR and sequence verified and delivered individually. Same as the pooled and representative library, each insert is cloned into your desired vector, and we can deliver up to 10,000 constructs. A final summarization of our platform, our combinatorial DNA library service is the one-stop shop for your metabolic pathway assembly needs. This service is highly customizable, meaning that if you have any other specific assembly requirements or quality control needs, we would love to discuss your specific case with you. Our current capacity allows to deliver seamlessly assembled inserts up to 15 kilobase in as fast as 4 weeks, which is unmatchable in the market. In conclusion, our platform is a faster and more economic choice for you compared to your in-house operations, thus can help speed up the building phase of your microbioengineering process.
Thank you, Dr. Li, for the wonderful talk. For more information regarding combinatorial DNA library assembly or our service, please visit our website at www.genscript.com, or you can directly email me at clairez@genscript.com. We'll start our Q&A session now. Please type in your questions in the question box if you haven't already done so. Finally, at the end of the presentation, you will receive a brief survey. This survey will only take you 1 minute. We strongly encourage you to fill out this survey, as this will help us better design our service as well as future webinar series to help you with your research. With that, we have already received some questions, and let's start our Q&A session now. The first question is, what do you think of using yeast assembly for building metabolic pathway?
Well, thank you for your question. I would say yeast assembly is very efficient. It is able to assemble up to 10 parts in one reaction, and it is able to construct very large DNA molecules to the genome-sized DNA molecules. However, a major disadvantage for the yeast assembly platform is that the manipulation of yeast cells is a little bit complicated, and most labs do not have this technique. And in addition, the yeast cells grow much slower than the E. coli cells, so it takes longer time for one round of assembly with yeast recombination. So the in vitro recombination is actually more preferred in the synthetic biology community. Yeah, that's the answer for your question.
Thank you. Let's see for the second question. The question is, if my design library is very large, can I still use arrayed assembly?
Thank you. The answer is definitely yes. But you need a lot of money and effort to test a very large library with arrayed assembly. But if you want to save your money and effort, a very good way is to use design of experiment to use your initial library size. This can significantly reduce your library size without losing much of the optimization effect. And this is especially powerful for multivariable testing. You can search the keywords of design of experiment to learn more about the strategy, and you can also find some relevant softwares to help you with the design. And I hope this is helpful for you.
Okay, let's go to next question. Third question, what's the best method for high throughput DNA assembly?
All right, best method, very straightforward question. I would say that every assembly method have their pros and cons. Take the Golden Gate assembly, for example, it is very efficient and it is very high throughput. The project I have described from Christopher Ward lab in MIT, they actually have used the Golden Gate assembly to build their combinatorial library. But you can only use this method only if your assembly can tolerate 4 base pair scars between the adjacent parts, and you have to avoid the enzyme cutting site in your fragment sequence. So this is the pros and cons for Golden Gate assembly. We here in GenScript we have our unique gene builder assembly method. It is based on in vitro homologous recombination, and it is very efficient. You can try to search the product in our website. But the best method, I would say, you have to use different methods for different projects. There are no method which is universally applicable for all projects. And well, I would suggest you to outsource your work to us, because we are professional on choosing the best way. Thank you.
Okay, we have a couple of more questions here. Let's take one more question. Here's a question. You mentioned to use combo assembly for protein engineering. Can you explain how does it work?
Yes. We do have customers who use our service for protein engineering or antibody engineering. They usually have different protein domain sequences with mutations. Maybe they have acquired their beneficial mutations in their previous rounds of protein engineering process. Now they want us to test different combinations of variant domains. You can also test the different combinations of the different CDR regions of the antibody, so that you can select better antibody variants. So that's the application of using our service for protein engineering.
Okay, thank you, Dr. Lee, for your answers, and thank you for the presentation as well. If you asked a question to us but didn't get an answer here, we'll send you an email to follow up on that. And thank you all very much for attending the webinar. We hope this is helpful for your research. And if you have any further questions regarding either the combo library or our service, please feel free to email me at clairez@genscript.com as shown here on this slide. Thank you all very much. Have a good day. Bye.
Bye-bye.